So we'll start with our next speaker. Nabusog po ba kayo sa lunch? Okay, good. So, Bean, can we play the introduction for our next speaker?
He's a scholar and researcher with a strong academic background and a passion for scientific exploration. He completed his Bachelor of Science in Biology from the University of the Philippines, Baguio, in 2008. As a Nova Ecija Foundation Scholar, in 2013, he pursued his Master of Science in Forest Products at Korea University, South Korea, under the Korean Government Scholarship Program. His quest for academic excellence reached new heights as he earned his Doctor of Philosophy in Molecular Biotechnology from Hiroshima University, Japan, in 2020. This achievement was made possible through the top global university project scholarship from METS Japan. His doctoral research focused on the microbiological synthesis of metalloid tellurium, with implications for biotechnology in heavy metal bioremediation and rare metal recycling. Following his undergraduate degree, he served as a research assistant at the R&D unit and molecular genetics laboratory at the Philippine Parallel Center National Headquarters in Cebu. After finishing his master's degree, he took on the position of instructor at the Department of Biology at the University of the Philippines, Baguio, building on his educational foundation to impart knowledge to future generations of scientists. Currently, he serves as an assistant professor at the Department of Biological Sciences at Central Luzon State University. In this capacity, he continues to shape the minds of aspiring scientists and engages in scientific research. His research interests encompass diverse fields, including bioremediation, molecular phylogenetics, and metagenomics. His work has led to several notable publications in prestigious journals, including Springer Nature. Some of his recent publications have explored topics like the discovery of a novel gene conferring telluride elements through telluride production and SGD-Yaku transformers in a marine sediment metagenomics. Additionally, his research has revealed 3 new marine material strains with telluride-reducing activity and tellurium recovery potential, isolated from Niigata. Our resource speaker, Dr. Madison B. Minor, Assistant Professor 4 of CLSU.
Again, let's welcome Dr. Madison.
So, good afternoon po to everyone. So, I will be presenting the discovery of novel genes from marine sediment through metagenomics. Actually, this is my dissertation in Japan. So, I will start my presentation with a brief introduction on the growing importance of tellurium, photoelectric and semiconductor metalloid used in photoelectric cells, solar cells, gadgets, computers. And this will be followed by a brief introduction on the 3 new bacterial strains isolated from the marine sediment, followed by the discovery of a novel gene through metagenomics. And I will end my presentation with a brief conclusion and recommendations. Okay, so tellurium is mostly recovered from copper mine tailings. The amount of extracted tellurium in a copper concentrate is generally below 100 parts per million. Significant increase in the market demand for tellurium encouraged some countries to explore other resources of this highly precious metalloid. So the top global producers of this metalloid are Europe, China, and Japan. Telluride is highly toxic to most microorganisms. So it generates superoxide radicals that interferes with biological functions of the cell, leading to cell death. Studies on microbial tellurium recovery reported several bacterial genera with telluride reduction and tellurium recovery potential, such as Staphylococcus, Bacillus, Cyclobacter, Rhodobacter, and Escherichia coli. However, most of the bacterial strains used in elucidation of the telluride reduction mechanism has very low resistance to the metalloid, thus hampering the understanding of the mechanism behind its reduction. For instance, Escherichia coli has a very low minimum inhibitory concentration of 4.5 micromolars in potassium telluride. Okay, so microbial cell-mediated telluride bioremediation and tellurium recovery offers a viable alternative as cleaner and sustainable technology to overcome tellurium resource scarcity. Tellurium is used in photovoltaic cells, rechargeable batteries, compact discs, as quantum dots for cancer diagnosis and treatment. Okay, so the marine sediment used in this study was generously provided by Dr. Takeshi Terahara, professor at the Tokyo University of Marine Science and Technology. So the marine sediment was isolated from Niigata Bay, Japan, 100 meters depth. So the culture showed a prominent growth of Maricromatium sp, a purple sulfur bacterium, as shown here with a purple pigmentation. And then it's a gram-negative bacterium known to be a telluride reducer. So the marine sediment was cultured or cultivated in RCBBN media. So with continuous elimination, since Maricromatium is a photosynthetic bacterium. And then, so the marine sediment showed a telluride reduction activity up to 10 millimolars sodium telluride. Initial identification of bacterial organism species present in the culture revealed the presence of Maricromatium, the Sulfovibrio, Pseudomonas enterococcus, Oceanotoga, Suzukinia, Vibrio, and Shawonella. Okay, so pore plating method was used to isolate culturable bacteria from the marine sediment enrichment culture. 3 distinct colonies were successfully isolated, and their genomic DNA was extracted, and the 16S RNA was amplified and was sequenced. So based on the blast homology search, so we revealed that the isolate 1 is Shawonella algae, isolate 2 is Pseudomonas alcaligenes, and isolate 3 is Pseudomonas itsuseri with 99% homology and an E value of 0.00. So phylogenetic analysis revealed that the isolated bacteria were not related to reported strains, thus new strains were indicated as Hero 1 for Shawonella, Hero 2 for Pseudomonas pseudocaligenes, and Hero 3 for Pseudomonas itsuseri as new strains of telluride reducing bacteria isolated from a marine sediment in Niigata Bay, Japan. Okay, so S algae Hero 1 and P itsuseri Hero 3, I should say, grew optimally at page 7, while Pseudomonas pseudocaligenes Hero 2 grew optimally at page 9. So the new isolates are marine mesophilic bacteria. The 3 new isolates are also facultative anaerobes. Optimum telluride reduction activity coincided with the optimum pH for growth. Gradual telluride reduction was observed in the 3 new strains. S algae Hero 1, this one, showed the highest resistance at 15 millimolars sodium telluride. The 2 Pseudomonas species showed growth and telluride reduction activity at 4 millimolars sodium telluride. Intracellular tellurium crystals were observed on the 3 new strains. Tellurium crystals in S algae Hero 1 showed needle crystals which annealed together forming T rosettes, possibly through electrostatic attraction. So for Hero 2, it showed small needle crystals And for hero 3, it shows TE nanorods. So both the hero 2 and hero 3, tellurium nanocrystals were scattered within the cell cytoplasm. So all are intracellular crystals. And the minimum unit size of the nanocrystals is about 16 nanometers. Okay, so that's for the isolation of the 3 new bacterial strains with telluride reduction activity. Now I will discuss the discovery of a novel gene using metagenome library construction and functional screening approach. So generally, this figure shows the general method on how to discover a novel gene through metagenomics. So metagenome DNA was extracted from the marine sediment using CTAB method. So here in the figure, I highlighted this one because this is the region where we get the DNA fragments, the novel gene, the gene that will be ligated in this plasmid vector. So both the genomic DNA was digested using SAL3A1 and the plasmid vector PHSG398 was digested with BAMH1. So these are compatible restriction enzymes. And then after ligation, so I did XCAL screening to screen the positive transformants. So out of the 20 positive transformants, there's one that showed positive for telluride reduction. Yeah. So and this was the insertion was confirmed using colony PCR using M13 primers. Thank God I have one. Yeah. All right. So sequencing the metagenome fragment with the telluride activity observed during the assay. So it revealed that there are 2 open reading frames. So one larger here starts here, or reading 1 with the red, and ends here, the stop, and the smaller ORF open reading frame here starts here, ORF2, and ends here. So there are actually 2 possible genes, ORF, that could be responsible for the activity, the observed activity. Okay, so using BLAST-N homology search, it revealed that the novel gene, the metagenome fragment, has homology with the PAP2 family protein with 92% identity and 0.00 p-value from Pseudomonas caesarea, one of my pure culture isolated from the same sample. And then BLAST-P homology search using non-redundant amino acid database, it showed that ORF1 has homology to PAP2 superfamily protein from Pseudomonas caesarea, and ORF2 has homology with phosphatase PAP2 family protein from Pseudomonas caesarea. However, it showed very high p-value. Okay, so comparing the sequence homology between the PAP2 superfamily protein and the metagenome fragment, we found out that only a fraction of the whole protein sequence of the PAP2 family protein was covered by the 2 ORFs in our metagenome fragment. So thus, the truncated sequence of these 2 ORFs may not retain the original function of the said protein. Okay, so predicting the possible biological and molecular functions of the ORF1, so it revealed a high probability of a substrate-specific transmembrane transport, both cation and anion and a possible catalytic activity. So this is just in silico. Alright, so phylogenetic analysis revealed that the A1 DNA, the metagenome fragment, was actually related to Pseudomonas caesarea. So we wanted to find out which of the 2 ORFs is the actual gene responsible for the observed activity, so we cloned both ORFs. So we used PTRC99A plasmid vector harboring a penicillin resistance gene. So the insertion here is at EcoR1 and V3. Okay, so the result of our expression, we revealed that A1ORF1 is the one responsible for the observed activity. Here in the figure, we show that the wild type carrying the negative plasmid without the insert, and the wild type competent cell, as well as the ORF2 containing expression vector had no growth, observable growth, and reduction activity. So ORF1 is the gene for the observatory. Okay, so tellurium crystals in the E. coli strain A1, we designated our transformant as E. coli strain A1 for tellurium crystals. These are not nanocrystals because it's more than 100 nanometers. So the minimum unit size of the tellurium crystal is about 500 nanometers, which is bigger than the pure cultures reported earlier. And the telluride reduction was observed to be gradual. So it took like days before you can see some blackening or the reduction process. So this result shows the optimum growth and telluride reduction activity of the three Pseudomonas, the wild type E. coli, the transgenic E. coli strain A1, and the expression E. coli. And then there's no significant difference between the optimum growth of the three. For the telluride reduction, so the optimum I think is 32 and 37. There's no reduction activity observed at 4 and 45 degrees Celsius. So the ideal optimum temperature for telluride reduction is around 32 to 37, and at pH 9 and 10, pH 7 and 9, and there is no telluride reduction activity observed at pH 10. Okay, so some insights on the bacterial tellurium recovery. So we did this using inductive decoupled plasma optical emission spectroscopy. So the three pure cultures show the highest recovery. So for the mixed culture, the whole marine sediment culture, harboring all the population of the bacteria, it showed 72% recovery. So among the pure cultures, Hero 2 showed the highest recovery, about 58%. And then there's no significant difference on the tellurium recovery of the three isolates at pH 9, below 50%. And the least recovery was observed in the transformant with 9% recovery, probably due to the short period of exposure, because it takes very long time for it to reduce the okay, so for the conclusion, so this study has isolated 3 new species of marine mesophilic bacteria with extreme resistance to telluride, which has the capacity to reduce and recover tellurium. And then we also reported a novel gene which confers telluride resistance through telluride reduction within the cells of the competent cells. So all these bacteria can be used for telluride bioremediation and tellurium recovery. So for future, protein isolation and crystallography might be of big help in elucidating the actual properties of the protein from the genome. So thank you for your attention.
Thank you so much, sir, for sharing your PhD dissertation with us. Q&A na tayo. Hindi, sir, tanong kayo sa anong gusto niyo po.
Questions for
Yes. Can we have questions from our participants? You can approach our ushers for the microphone po. If wala pa, ako muna, sir, may tanong. Yeah. So, you mentioned, sir, na yung telluride is toxic to various creatures such as, especially to bacteria, tama po ba? So, ano po yung nakikita natin application in terms of disease prevention or cure in aquaculture?
Ayan, so medyo off tangent kasi yung yung presentation ko. So medyo wala about aquaculture. So yun lang, medyo marine lang yung example ko. Kaya more on bioremediation and metal recovery yung research.
Ano sir, for your marine?
For possible application siguro for bioremediation ng water pollution.
So
In general, if you can use this.
Magagamit din po natin siya sir sa plants, for example.
If ever may contamination ng meta,
Ay, opo. Yes, sir. May mga ano kayo, sir, further studies na pinapursue about these, yung nawa? Sa
for now, wala
reproduction.
pa. But meron, may some part of study. Meron akong isolate na mutant na hindi kasi na-sequence yung mutation, so hindi pa siya napapapush. So I'm looking forward to continuing that part of the research. Kasi mas tumaas pa. Ayan.
Do we have other questions from our participants? Last chance na natin magtanong sa ating mga speakers. Though after, pwede naman atang matap sila kung may additional questions pa.
Simple question. Yung sa annotation, anong ginamit dun sa silico? So, para malaman yung predictive function ng gene. So, cross annotation lang ba yun? Or gumamit kayo ng tools para infer yung possible function?
Okay, so thank you, ma'am. So I think the accepted in silico prediction of protein structure, biological function, molecular function is gene ontology. So among the in silico analysis that I perform, that one is the only accepted by my reviewers, gene ontology. So that's the most probable function of the protein isolated in silico.
Did you also do the structure prediction?
Yeah. So actually, yung AlphaFold, which is the very recent structure prediction software, I tried it using. But before the AlphaFold, I also tried predicting the structure of the protein. So it's a transmembrane. There is a 6 helical transmembrane. Most probable structure of the protein from the genome. So based on the AlphaFold prediction, somehow it agrees with the number of alpha helices from the sequence, amino acid sequence. But unless you perform crystallography, that's the only time you can say that this is the actual structure of the protein. So I can still say that.
Okay, thank you, ma'am. So, some other questions from our participants?
Hello, good afternoon po. I'm Samuel Ian Mohino from Cavite State University. Pero dito rin po ako graduate sa CNSU. So yung word po na metagenomics is palagi ko pong naririnig. And sa totoo lang, hindi ko maintindihan kung ano po talaga ang metagenomics. Kamay ko lang siya naririnig pero hindi ko totally maintindihan. Tanong lang po sa mas madaling sabi or ano po ba talaga yung, ano po ba ang metagenomics po? Para sa mas madaling maintindihan po. Thank you po.
Okay, so metagenomics, it's a tool, it's a technique of extracting DNA without culturing. So it doesn't entail a pure culture. So in short, environmental DNA, direct from the sample. For example, if you have a marine sediment, you're going to extract the DNA out from that sample, environmental sample. So you don't have to purify specific organism to be able to extract the DNA, the genomic DNA. So the DNA extracted from a metagenome or environmental sample is called a metagenome DNA. So the metagenome DNA is summation of all the organisms. For example, in bacteria, all the population of community of bacteria present in the environmental sample. So it doesn't entail culturing. So you are going to capture all different microorganisms present in the sample.
Thank you very much po. At follow-up question po. So, pwede pong ma-detect yung environmental DNA from any virus, for example, sa water. So, kahit wala na po dun yung organism na yun, pwede pa rin pong ma-detect na possible na dati may ganong organism po.
Yes. Actually, the reason why we have metagenome is because of the culture-dependent lab in microbiology. You need to have the media in order to culture specific microorganisms. However, in reality, we can only culture 1% of the bacteria present in the environment because of we lack the appropriate medium to provide or nutrition or environment to grow them in the laboratory. That's why we have this metagenomics. So we can actually capture those unculturable bacteria from the environment because we are lacking the mechanism to grow them in the laboratory.
Thank you very much po.
Thank you po. So any other questions from our participants?
Nadagdagan ko yung question. So, how will you know what are those organisms that are unculturable from the metagenomic data that you get?
Okay, so sa process for metagenome DNA library construction, you're actually going to create a library. So you're going to fragmentize all the genome present in the sample. So all the fragments generated will be cloned in a plasmid, and later those fragments will be sequenced. So that's the time that you're going to know the identity of the organisms present in the sample. So we're going to make a library, metagenome library or cDNA library. It's the same thing. However, for metagenome library, it's different DNA from different microorganisms.
Have you identified, have you identified any of the microorganism or organism from the metagenome that you collected?
If you have the library, you can sequence all the colonies to get the identity of all the organisms present in the sample. So in our case, we are just up to screening a gene with a specific activity, which is the reduction of the meta. So.
Thank you, ma'am.
So just a clarification, dun sa pinakita mong phylogenetic analysis kanina, it is just based on the gene or you already have the whole sequence?
Sa A1 fragment po, sa metagenome
You mean
fragment.
year 1, year 2, year 3?
Sa isolated bacteria po. So this actually almost full complete sequence ng 16S RNA. Yung basis po namin. So these are pure cultures.
So complete na siya?
Yes.
So kung complete na siya, ma-infer mo rin yung gene na pinufocusan niya for the specific function for Tellerite. Would you know kung nasa ang operon siya na encode and ano yung mga genes na katabi niya or pwedeng related sa function niya din?
Based on our BLAST-P analysis, if you feel that it's closely related to PAP2 superfamily protein, that's why I made some alignment. So it showed, based on the alignment, only a fraction of the PAP2 superfamily protein is covered by our metagenome fragment. So we infer that the truncated sequence may not be able to retain the actual function of the protein with homology.
Thank you, Doc. Sir, sinabi niya sa bioremediation.
Yes.
So, removing pollutant from, for example, sa pan. So, paano natin yun, sir, mapoproduce yung sa genes? Paano yung process niya? Paano natin siya papadami?
Okay, so we have competent cells. So these competent cells are the ones carrying the gene expressing the protein which can bioremediate the metal. So for instance, here, it can bioremediate. It can turn the toxic telluride into elemental form, which is tellurium. And tellurium is a highly valuable metalloid. It's a semiconductor and photoelectric metalloid which can be collected can be used for solar cells and other gadgets.
Ano sir? Economical ba siya sir? Or would it
Yes, because the traditional, conventional method of remediating toxic pollutants entails high energy and it uses toxic chemicals. So instead of using toxic chemicals to remediate these metals, you can use microorganisms instead to bioremediate these toxic.
and it won't have any potential risk naman to
Potential risk, so E. coli might, pero I think best muna in a controlled environment. You can get the polluted sample and then bioremediate it in, hindi ko pa na-envision na in situ. Kasi pag in situ, madaming considerations. You may pollute yung environment using microorganisms, the E. coli, the competent cells. Yun yung concern.
Yeah. Future, it's future works. Yes. I think okay na po tayo. Is there any more questions? Okay na po. If may maalala kayo mamaya, pwede niyo po i-approach si Sir. Yeah. So, thank you very much, Sir. Yeah. Kay Aljun.
Thank you po, Dr. Monar. Medyo knowing lang po yung ating mga participants siguro sa metagenomics since nga ngayon po. That's why we have this kind of symposium para po malaman po ninyo na may ganito pong application po. And maybe in the future nga po, mas madami pa po itong application po for fisheries and aquaculture. So again, thank you, Dr. Madison Monar. And now, we would like to award the Certificate of Appreciation to Dr. Madison Monar for sharing his valuable expertise and knowledge as resource speaker during the 8th National Fisheries Biotechnology Symposium with the theme Empowering Innovation for Sustainable Future with Biotechnology in Aquaculture and Fisheries, awarded on November 22, 2023 at BIFAR NFDC, CLC Compound Science City of Muñoz, Nueva Ecija. Signed, Dr. Lillian C. Garcia, SF5, Executive Director, DANF RDI. Let's give him a round of applause.